All right, now the chapter on cell membranes. Cell membranes are so important, they come up in pretty much every lecture we've done so far in one way or another. Now we're going to actually focus on them and what they do, what they're composed of. So a few things about cell membranes. They are selectively permeable. That means that some things can get in and other things cannot. So some things can permeate, right? Some things can get in and out, and some things have a harder time getting in and out. And they're composed primarily of phospholipids, a molecule that we'll talk about in an upcoming lecture. But these are amphipathic molecules. Amphipathic molecules, let me draw one here. This is a phospholipid. It has a phosphate group here, which has a negative charge, and then it has fatty acid tails. So when a molecule is amphipathic, that means that parts of it are polar, also, which makes them hydrophilic. Okay, hydrophilic, that means they like water. Now these tails down here are made of fats, right? And these are nonpolar, which means they're hydrophobic. So these molecules have parts that can interact with water and parts that are repelled by water. And that's what amphipathic means, okay? Here's the structure of one of these things in more detail. You can see the fatty acid tails here. You can see the phosphate group up here. I usually draw them like this. And they have a hydrophilic head and a hydrophobic tail. Hydrophilic means they like water. Hydrophobic means they don't like water. Now, if you place these into a cup of water, one thing that might happen, here's the water, right? So one way these might arrange themselves if they were placed in water would be that the hydrophilic head would be in the water and the tails would stick out. But they could also form a sphere, a shell, if you will, some kind of sphere like that. And that's one thing that could happen, and that can happen, but the way that biology works, the way that these work in a membrane is they make a double layer. So they make a sphere of these things, but it's not just one layer thick. So let's say you have a cell in a cup of water, right? There's water out here, and there's water inside the cell, right? I mean, there's liquid in the cell and outside the cell. So how do these things work if parts of them like water and parts of them do not? Well, here is one of these things here, right? So this is how they would do this, a double layer. And it would be all the way in a circle, all the way around in a sphere, but I'm not going to draw the whole thing. Like this. They would have the hydrophilic parts facing water, and the hydrophobic parts would be together in the middle in an area that excludes water. And this would be called a lipid or a phospholipid. What am I doing? I can't write and talk. A phospholipid bilayer. Bilayer. And that's what we see here. Phospholipids form this bilayer that forms the whole membrane. The area in the middle excludes water. There's no water in this area here. This is the hydrophobic area. And then there's the hydrophilic heads facing water. Phospholipid membranes are made up of way more than just phospholipids, however. Outside of this membrane, of course, we have the extracellular matrix, which is stuck to proteins that are embedded in the membrane, and those proteins might also be connected to the cytoskeleton, like we see here. So we have proteins that cross the membrane. These proteins that cross the membrane are known as integral proteins, and they also are amphipathic. They have polar parts up here, or hydrophilic parts, right? And then the middle part here is hydrophobic. So they are structured in a way where the amino acids that like water are towards the outside. The amino acids that are repelled by water are folded towards the inside. There are also proteins that might be stuck to the outside of the membrane, known as peripheral proteins. And we also see other structures here like glycolipids. That's a combination of a lipid, a fat, and a sugar. like glucose, and glycoproteins, which are proteins with sugars attached to them. We can see those stuck to the membrane. So there's a lot of stuff going on in a membrane. And one other part you might find, at least in an animal cell membrane, is this molecule here, cholesterol. Cholesterol has a really important function in animal cell membranes. So there's several parts. The primary part are phospholipids and proteins, but there are other components that we should be familiar with as well. Now, these membranes follow something known as the mosaic, I'm sorry, not the mosaic, but the fluid mosaic model. This is the way we think membranes work. What this means is that think of it like an ocean of phospholipids, and the proteins on the surface are like icebergs, and they don't just sit in one spot. It's fluid. Things move around. So it's said to be like a mosaic, like if you think of a picture that's a mosaic made of little pieces, but those pieces can drift around. Those proteins can drift around. And the way they kind of came up with this idea, it used to be thought that everything just stuck in place, but they took a mouse cell and they looked at the proteins in the mouse cell, and they squished it together with a human cell that have different proteins on them. And they looked at it after about an hour, and the proteins were all mixed up. So these proteins don't sit still. They drift around inside of this membrane. They move around a little bit. And in fact, what we found out is if you make a membrane less fluid, it doesn't work very well. So what happens when it gets cold? Well, as we'll learn in greater detail when we talk about chemistry, but I'll show you here. Membranes change their fluidity. When it gets really cold, membranes could solidify. They could get hard. Just like when you put some soup or something that has a lot of fat in it in the fridge and the fats harden up. So if there's a lot of saturated fats in the cell membrane, they pack together and they become a solid when it gets cold, and that would be bad for the cell. So let me draw you. This is just a phosphate head with fatty acid tails. Saturated fats have straight tails on them like that. And when they get cold, they can pack really tightly together. And that would be solid. Unsaturated fats, however, have tails that often have bends in them like this. And so they can't pack tightly together because they have these bends that bump into each other. And so these stay liquid. Sorry about that. These stay liquid at colder temperatures. And so this is one way that a cell membrane might avoid getting too tightly packed together if the organism lives in an environment where it can get chilly. Another way to do this is you could still have membranes made of saturated fatty acid tails, but then you stick cholesterol in there, and cholesterol can keep things from packing too tightly together. Let me clear the drawings off of this. Here we go. So if you have cholesterol, it restrains the movement of phospholipids at warm temperatures, which means they don't move too much. Because if you have a membrane and you get it warm, these lipids move a lot when things get warm. Remember, heat energy is random motion, and so if these things move around a lot, you get too liquidy, and that's bad too. Your cell would kind of fall apart. So cholesterol restrains movements at warm temperatures, and when it's cold, it prevents things from packing together too tightly. So cholesterol is the way that animal cells usually deal with this. A lot of plant cells will have more unsaturated fats, and I think bacteria cells may do that as well in their cell membranes to prevent them from packing too tightly together at cold temperatures. Now, some bacteria actually have an enzyme called fatty acid desaturases, which allows them to change from saturated fat to having at least one of these tails be unsaturated, where it has a bend in it. And we'll talk about why there's a bend in that when we get to the chapter on chemistry soon, if you haven't learned it already when you're watching this video. So here's some examples of this. Here's a cell membrane with a lot of unsaturated hydrocarbons, and this, even if it was cold, would still be pretty fluid. Whereas in this case we have saturated tails. They're all straight tails. They're packed tightly together. This would be more viscous or thick, right? That's what viscous means. And in animal membranes, we still usually have these saturated tails, these straight tails like we see down here, but we pack cholesterol in between a lot of them that prevents them from packing too tightly together. But it can also keep them from moving too much when it gets warm, preventing them, the membrane from becoming too fluid. All right, so in the next video, we're going to continue with this and we're going to introduce membrane proteins. You've heard a lot about these probably already, but we're going to introduce them formally in the next lecture video.